So next one is a cell membrane. Cell membrane is a structure which is present just below the cell wall. If you see the bacterial structure, so this is a cell wall which is present outside that one. And here below this one the plasma membrane is present, which is made up of lipids and proteins, as like that of a normal plant cell. The cell membrane is made up of lipids and proteins, which is considered to be the living limit or living membrane, and which is selectively permeable property. So what do we mean by the selective permeable property means it will allow only some few number of solute molecules into the cell, and that is the way how it is selecting only particular solutes which are useful for the cell. So apart from this structure, the cell membrane has some special structures. Here we can see some invaginations. Here and there we can see some invaginations inside the bacterial cell. These invaginations are called as mesosomes. So these are the special structures which are considered to be the structures where the absorption area is increasing, and it is helping in the replication and distribution of genetic material between the daughter bacteria during binary fission. And initially some of them considered this as a site of respiration also. So likewise, why they consider it as a site of respiration means because in the bacterial cell the respiratory organ, that is mitochondria, is absent, and that function is taken up by the mesosomes, because the mesosomes contains the enzymes of respiration and also the electron carriers which are required for the process of respiration are present in the mesosomes of the plasma membrane. So likewise, these are some special characters which are being identified in the mesosomes, which are present only in the bacterial cell. In these mesosomes, in some of them they are considered to be the septal mesosomes, and some of them are lateral mesosomes. Actually, the mesosomes which are having the respiratory enzymes or respiratory related aspects, so such type of mesosomes are called as a septal mesosomes, and lateral mesosomes are the one which lack respiratory enzymes in them. So based on this character, they had differentiated the mesosomes into the septal mesosomes and the lateral mesosomes. And during the process of binary fission, the bacterial cell will divide, and the site of division will be the mesosomes, and it not only help in the division but also helping in the replication of genetic material and distribution of the genetic material between the daughter bacteria cells. The normal functions related to the cell membrane are: one is a selective permeable property, selective permeability, and apart from this one, it also help in the process of osmoregulation, that is maintaining the concentration of the water inside the bacterial cell. So this is about the organization that can be seen in the cell membrane. And the other contents which are present inside the cell membrane is called as a cytoplasm. The cytoplasm is a fluid content which is present inside the plasma membrane, and this cytoplasm includes major portion which includes the water, and some cell organelles are present like ribosomes. Here the ribosomes are of 70S type of ribosomes are present, and as we know that every ribosome contains larger subunit LSU and the smaller subunit. Here in the 70S type, the larger subunit is 50S and smaller subunit is 30S. And generally sometimes whenever the process of protein synthesis is taking place, the ribosomes will be in the form of a chain-like structure. This chain of ribosomes is called as a polyribosome or the polysome structure. Polysome or polyribosome. Here the polysome or polyribosome is formed with the help of a mRNA molecule. That is, mRNA will hold all these ribosomes in the form of a chain, and once the protein synthesis have been completed, once again all the ribosomes, that is smaller subunit and the larger subunit, will be separated from each other, and mRNA will be helping in detachment so that all the parts will be separated out when protein synthesis is completed. So apart from the ribosomes, what are the other cell organs which are present in the bacteria means, here generally it does not have, it lack membrane bound cell organelles. So some cell organelles have double membrane and some will have the single membrane. Double membrane cell organelles like mitochondria, chloroplasts, and single membrane cell organelles like Golgi apparatus or Golgi complex and endoplasmic reticulum. All these cell organelles are absent in the bacteria. So as a mitochondria is not present, the aerobic respiration is not possible, and chloroplast or the lamellar structure which are not present. But there are some bacteria which are photosynthetic in their nature. Those bacteria will have some lamellar structures, so which are not exactly as like that of chloroplast, but performing the function of a chloroplast by having the pigments in them, that is bacteriochlorophyll or the photosynthetic pigment present in them. And Golgi complex and endoplasmic reticulum are completely absent in this bacterial structure. Apart from this one, here it have the double-stranded DNA containing structure. This is called as a nucleoid or the genophore. It is considered to be the primitive nucleus. Why? Because the nucleus lack nuclear membrane, nucleolus and nucleoplasm. The nucleus or the genetic material, we can say, better to say it as a genetic material. The genetic material is represented with circular double-stranded DS, double-stranded naked DNA. So which is freely present in the cytoplasm without any boundary. So this type of DNA or genetic material is called as a nucleoid or the genophore. And apart from this one, there is one more structure which is present in the cytoplasm, that is called as a plasmid. Here the plasmid is the extra chromosomal DNA. So this extra chromosomal DNA, it is present in the cytoplasm and it can replicate by itself, that is, it is self-replicating. It is independent of the replication of the DNA present in the nucleoid. But sometimes in some of the bacteria, the plasmid may get associated with the DNA of the nucleoid also. When it is associated with the DNA of the nucleoid, along with the DNA of the nucleoid, the DNA present in the plasmid will also start replicating. but when it is independently present, it can replicate by itself without the help of the nuclear or nucleoid DNA material. So here the plasmid is exploited in genetic engineering. In which manner it is exploited means it is used as a vector for transfer of desirable genes from one organism to the other organism. So it is acting as a vehicle for the transfer of genetic materials or the desirable gene from one organism to other organism in the technique of genetic engineering. So why it is exploited for that purpose is the plasmid is made up of few number of genes and majority of the functions are not much useful. So that's the reason why it can be exploited for using as an vehicle in the transfer of desirable genes from one organism to the other organism. And plasmids are of various types based on their nature. So let us see what the three types of plasmids are present in different types of bacteria. Those are F plasmids. F plasmids are the one which are responsible for fertility, whereas we call it as a F factor, and the bacteria which contains this F plasmid are called as F plus bacteria, and the one which are not having this F plasmid are called as a F minus bacteria. And during the process of conjugation, the F plasmid will replicate, and when it is replicated, it forms an extra additional plasmid. That plasmid will be transmitted from F plus bacteria to the F minus bacteria. And second one is the R plasmid. R plasmid is the one which is providing one special characteristic feature, that is resistance against antibiotics like tetracycline, chloramphenicol, ampicillin, etc. So this is a special biological character that is acquired by the bacteria due to presence of this R plasmid. The other one is the col plasmid. So the col plasmid is the one which contains or which are called as cosmids, which have one toxic factor that is called as a col. That's why we call this as a col plasmids. So it is poisonous or toxic to the bacteria, which is causing harm to the particular bacterial species. So this is a way how these different types of plasmids are there, and their function is also been acquired based on that nature of the genes which are present in the particular plasmid material. Majority of the bacteria are heterotrophic in their nature, but some of them are autotrophic also. So the one which are autotrophic, which will perform photosynthesis, and such bacteria are called as a photosynthetic bacteria, and these photosynthetic bacteria contains some special structures which are having the photosynthetic pigments in them. The photosynthetic bacteria like green sulfur bacteria, that is chlorobium, and the other one is purple sulfur bacteria. It is called as chromatium. Here the green sulfur bacteria will contains lamellar structures below the cell wall. These lamellar structures will contains the pigments which are responsible for photosynthesis, whereas the purple sulfur bacteria will contains invaginations from the plasma membrane. Those invaginations will contains the photosynthetic pigments which are required for the photosynthetic process. So here the lamellar structures are called as chlorosomes. and here the invaginations are called as a chromatophores. That's what the variation can be seen, but both of them are helping in the photosynthetic process with the help of the pigments. The pigments which are present in this bacteria are not chlorophyll A and chlorophyll B as like that of the normal plants, but they have special pigments are called, those are called as a bacteriochlorophylls. Some bacteria have some inclusion bodies in them. So apart from these inclusion bodies, they have some of them will have the gas vacuoles. These gas vacuoles are membrane bound, and these gas vacuoles are concerned with the buoyancy, that is ability to float on the surface of the waters. So here these gas vacuoles are permeable to gases only, but it is impermeable to the water, and it is little bit concerned with the osmoregulatory function also. So this type of gas vacuoles can be seen in aquatic bacteria like halobacteria. The inclusion bodies are there which contains vesicular structures. Those are called as sulfur vesicles, iron vesicles, phosphorus vesicular structures, so which are helping in the storage of those particular substances. These structures are granular structures. These are called as sulfur granules, iron granules, phosphorus granules. Some will have magnetite granules also. So these granular structures are considered to be the inclusion bodies, and when this food material have been acquired by the bacteria, it will store the food materials in the form of reserve food materials. So it will be in the form of polyhydroxy butyric acid, or we can call beta polyhydroxy butyric acid, and some will in the form of a glycogen molecule. So glycogen is a type of a polysaccharide molecule. So likewise the reserve food material will be stored in the form of poly beta hydroxy butyric acid or the glycogen granules. Next characteristic feature is the type of nutrition that is exhibited by the bacteria. As we said that basically bacteria will exhibit two types of nutrition. One is autotrophic and other is a heterotrophic type of nutrition. So as we know that autotrophic means which can perform a process where they can prepare their own food materials, whereas the second one is a heterotrophic type of nutrition. Actually here the nutrition is divided into two types in bacteria based on the source of carbon, that is carbon dioxide, and the source of energy. That means from where does they are getting the carbon dioxide and from where does they are getting that energy. Based on that one they have been divided into autotrophic and the heterotrophic type of nutritions. So let us start with the first one, autotrophic, which can prepare their own food materials by performing either photosynthesis or chemosynthetic process. So here let us start with the first one, photoautotrophs. Here photo means light, that is the source of energy in this bacteria is light, and the source of carbon or carbon dioxide is inorganic carbon dioxide from the atmosphere. So this type of bacteria are called as a photoautotrophic bacteria. Those are green sulfur bacteria, that is chlorobium and purple sulfur bacteria, that is chromatium or chromatium. In the autotrophic, second one is the chemoautotrophs or chemoautotrophic type of nutrition. In the chemoautotrophic type of nutrition, the source of energy is oxidation of inorganic chemicals. That is, when the inorganic chemicals are oxidized, they will release some energy. That energy is useful for performing photosynthesis. And the source of carbon is the carbon dioxide from the inorganic carbon, that is carbon dioxide from the atmosphere. So, based on that type of chemical which is oxidized, so here they are further divided into different types, like nitrifying bacteria. Based on the type of the inorganic chemical that is oxidized. So, the chemoautotrophs have been divided into, one is nitrifying bacteria. So, the nitrifying bacteria which will oxidize the nitrogenous compounds like Nitrosomonas and Nitrococcus. Here it will oxidize the ammonia molecule and convert it into nitrite with releasing some energy. And other nitrifying bacteria are the Nitrocystis and Nitrobacter. So, these are the bacteria which will oxidize the nitrite further into nitrate molecule by releasing some energy. That is, when ammonia is oxidized, it is releasing some energy, and when nitrite is oxidized, here also it is releasing some energy. This energy is utilized by the nitrifying bacteria as a source of energy, or the energy which is required for that process is taken from this oxidation of this ammonia or the nitrite molecules. Based on the type of chemical that is oxidized, the other type is the sulfur oxidizing bacteria. The sulfur oxidizing bacteria, one of the example is Beggiota. So, Beggiota is a bacteria which will oxidize hydrogen sulfide and convert it into molecular sulfur by releasing some energy. And one other is there, that is Thiobacillus thiooxidans. This is a one which will oxidize the sulfur molecule and release sulfates along with some energy. That is, here the sulfur, elemental sulfur is converted into sulfates. When this is taking place, here also some amount of energy will be released, which is used for the photosynthetic process. So, the other type of chemoautotrophs are iron bacteria. So, here the example for one is Ferrobacillus ferrooxidans. So, this is the one which will convert the ferrous compounds into ferric compounds. So, when these ferrous compounds are converted into ferric compounds, here also some amount of energy is used is released, which is used in the photosynthetic process. Likewise, based on the type of inorganic compound which is oxidized, the chemoautotrophs are divided into various types. Then next one is the heterotrophic bacteria. In the heterotrophic bacteria, are the one which cannot prepare their own food materials, but they depend upon others for the nutrition. So here these are of two types. One is called as a photoheterotrophs and other is a chemoheterotrophs. Here in photoheterotrophs, the source of energy is sunlight, whereas the source of carbon is organic compounds which are present in the other organisms, that is a host. In the photoheterotrophs, examples are Rhodospirillum, Rhodomicrobium, like examples. And when it comes to the chemoheterotrophs, so these are the one, the source of energy and the source of carbon, both of them are coming from the organic compounds. That is, these are the one which are in various manner, like saprophytes. Saprophytes are the one which will depend upon the dead and decay organic materials. That is, they derive the nutrition from the dead parts of the plants and animals. The best examples for this one is the Bacillus species. And in chemoheterotrophs, the second type is the parasites. Parasites are the one which will derive the nutrition from the living host, and they are causing damage to the host organism on which they are depending. So here the examples are Salmonella, and the third type is the symbionts. Symbionts are the one which are showing a mutual beneficial association where both the host and the dependent are benefited by this association. So this type of nutrition is called as a symbiotic type of nutrition, and organisms are called as symbionts. The best example for this one is a Rhizobium bacterium. The Rhizobium bacterium is associated with the roots of leguminaceae plants, where the rhizobium will provide nitrogen to the legume plant, whereas the legume plant will provide nutrition and also shelter to the Rhizobium bacteria. Likewise, both of them are benefited with this association. This type of association is called as symbiotic type of association, and this type of nutrition is called as a symbiotic type of nutrition.